Alright, okay, so hello dears. Ayan, so welcome back to our pre-recorded lecture on anaerobic culture. Alright, and for this video, we're going now to discuss, or we're going to start discussing on the bulk now of the culture mismo, which is again specimen processing. Okay, alright, so how do we process our specimen for anaerobic culture? Okay, alright, so first one, our general procedure is of course, if you receive a sample for anaerobic culture in the laboratory, of course, if wala siya nabutang in anaerobic transport, medium daan or anaerobic conditions pag-transport, then as much as possible, idiretso niyo siya butang og anaerobic chamber or an area na wala gyoy, or an area na naa sa in anaerobic environment. Okay? Alright, because again, we want to limit and we want to avoid as much as possible the introduction of or the presence of oxygen. Alright? Para the next succeeding steps on processing the specimens will be done on in an oxygen-free environment. Okay? Alright. First is of course macroscope examination of the specimen. So your specimen you observe again for any granules ba, diba? Sulfur granules, na ba siya odor, daan, etc. etc. Unsay color sa fluid or unsay color sa tissue, mga inana. So you observe macroscopically, of course, first, and then next you perform gram staining. Now for anaerobic culture or for anaerobic organisms, some of them do not accept or they do not hold on to the safranin that well. So pwede tang mo make og counter mechanisms or modifications for that. So we can use again 0.1% basic fuchsin, or we extend the time for safranin to 3 to 5 minutes para mas ma-ensure gyud na mas ma-take into nila ang safranin, or because again some of them they do not take well the safranin. Okay, so pwede nato siya extend ang time, or we use another counter stain that is 0.1% basic fuchsin. And of course, fixation, methanol fixed siya, dili i-heat. Alright, methanol fixed. Okay, next, inoculation of appropriate plated and tube media. As I mentioned sa culture media prep nato na lecture, as far as I can remember, I mentioned na your anaerobic culture, anaerobic organisms, they have their own set of culture media. Although most of them, some of them are like same lang sa normal routine culture media, naagi uban na specific for them, or like ginagamit lang yun siya for anaerobic culture. And we'll go into that sa next slides, alright? So your culture media may contain vitamin K, hemin, and yeast extract, again depending on the needs of the organisms. And some of these are what the anaerobes or your anaerobes need. Alright, okay. And anaerobic incubation, again, since we need to maintain an environment that is free of oxygen, dili gyud nato siya lahi iyang incubation specifications or requirements. Dili siya same sa atong normal na incubation, of course. Because again, our normal incubation contains a lot of oxygen, or it is an environment that has oxygen. Now for anaerobic culture, dapat gyud walay oxygen. Ang iyang incubation. Okay, so we'll go into that later. Alright, so first we'll go into the inoculation procedures, alright? And we'll start first with the primary plating media. So what are the culture media na pwede natong gamiton as primary plates for our anaerobic culture? First is of course blood agar, pero anaerobic lang siya. So it's just a normal blood agar, pero anaerobic siya pag incubate. Okay? So it supports the growth of almost all anaerobes, and it's best for anaerobic gram-positive cocci. So again, it's just a normal blood agar, like ginagamit nato for your normal blood agar, pero gina-incubate lang siya at anaerobic conditions, okay? So enriched medium containing sheep blood for enrichment and detection of hemolysis. So still the same, all right? So with the normal routine blood agar na ginagamit. But again, the point lang is incubate siya at or in an anaerobic environment, okay? All right. Next, you have what we call the BBE, or Bacteroides bile esculin agar. Dili BBC ha? Dili big black char lang. BBE, Bacteroides bile esculin agar. So with the name itself, sa agar, para kakinsa siya, it's for Bacteroides, which is one of the most common anaerobes na ma-isolate from clinical specimens. Okay. Now, again, this is a specific agar for or culture media for anaerobes. Dili niya siya ginagamit under normal kanang plating media for aerobic organisms. Again, it's usually reserved for anaerobes. So the Bacteroides bile esculin agar, again, isolation of Bacteroides fragilis group, as I've mentioned the most common isolated anaerobes in our clinical specimens, okay? It contains gentamicin, 20% bile and esculin because your Bacteroides fragilis is bile resistant, okay? So, ma-resist niya ang bile, okay? Alright. And mo hydrolyze mo siya esculin. So, if na blackening, it means iyang gi-take up ang esculin. So, esculin hydrolysis positive. Blackening of the medium. Alright, if I'm not mistaken. I remember. Okay, alright. Next, we have your Brucella blood agar. Still the same blood agar that supports the growth of almost all anaerobes. And it's suitable daw for anaerobic gram-neg bacteria. Okay? Alright. And still the same. Actually, same na siya with the blood agar pud na anaerobic, no? Pero kani siya, it's, I think there are some enrichment pud na gi-add aside from sheep blood. Okay? So that's the Brucella blood agar. Alright. Next, you have the kanamycin, vancomycin late, okay, or LKV. LKV na siya na, or KVL ba? KVL blood agar. So, what it does is, it's late. When you say late, the blood agar, or the red blood cells, or the blood, underwent, or has undergone repeated freezing and thawing. So, like, gipagahi siya, gi-freeze, tapos gipamelt na po, then gipagahi, gipamelt, gipamelt, yes naman. Alright, so freezing, thawing, repeated freezing and thawing of the blood para mulize daw ang cells, okay? So, blood agar yung base, tapos gilake ang blood para again mulize ang cells. So, repeated freezing and thawing, alright? And giadan og mga kanamycin, vancomycin, which are antibiotics. Okay. Alright, so contains kanamycin, vancomycin, and late blood. Again, late, when you say late, late is repeated freezing and thawing of the blood para mulize ang RBCs. Okay? Alright, and it accelerates the pigmentation by certain Prevotella species. Okay. Alright. Next, we also have egg yolk agar. It's used to detect lecithinase and lipase enzyme capabilities of Clostridium and Fusobacterium. So, usually, sa lecithinase and lipase, your Clostridium dyan ang well-known for that. Like, I think Clostridium botulinum is lipase positive, as far as I can remember. Okay? And some Fusobacterium. So, egg yolk agar, to detect the production of these enzymes, lecithinase and lipase. Okay? And also PEA, phenylethyl alcohol. It supports the growth of almost all anaerobes, contains sheep blood cells, and phenylethyl alcohol, which inhibits gram-negative and swarming of proteus. Okay, alright. So, PEA, phenylethyl alcohol. Alright. Okay, next you have also CCFA or cycloserine cefoxitin fructose agar. Diba? Sa stool culture na mention ni siya, it's selective yun. So, ang mutubo ra yun ana is Clostridium difficile or difficile. Clostridioides na siya karon. Okay, Clostridioides difficile with characteristic yellow ground glass colonies and unsa to na odor, horse stable, animao siyang kabayo. Alright, okay. Contains cycloserine cefoxitin, which are again antibiotics, and neutral red, okay, which is your indicator. Okay, and you have the cholestynalytic acid agar, di ba? Na-mention na to sa culture media prep. It's for specifically for gram-positive, di ba, organisms. Inhibits the gram-neg bacilli and still proteus. Okay. Alright. Now, we also have the Thiogl Glycolic broth. Usually, your thio broth is not only for anaerobes. Pwede po siya magamit for aerobes. Basically, it's an all-around broth. Actually, pwede siya magamit, no? Pero, for anaerobe purposes, no? Again, non-selective siya for anaerobes. Pwede po for aerobes. Okay? Na siya Thiogl Glycolate and agar, potential, agar, yes, lower oxidation reduction potential, glucose, and may be supplemented with hemin and vitamin K. And again, di ba, remember, unsa tong na-mention ako sa intro to micro na indicator sa oxygen, ani, you have risazurin. Okay? Remember? Okay? And risazurin, if naay oxygen, oxygen is present, color pink, mahimong pink ang Thiogl Glycolate broth. And when it happens na mahimong pink ang broth, Thiogl Glycolate broth, di ba? You boil, di ba? You boil the broth para mawala tong oxygen. Okay? Alright, so muna siya. Thiogl Glycolate broth. Again, all around, pwede siyang aerobes and anaerobes. Alright. And we also have the cooked meat, medium, or broth. So I'm not sure unsa na meat, okay? Pero again, still the same. It's for your anaerobic needs, alright? So, and chopped meat, non-selective for cultivation of anaerobes pa rin. So the meat serves as a source of protein nila, no? Peptides, and all the other nutrients. Okay. Meat particles initiate bacterial growth and reducing substance. So, na mga reducing substances na mo-lower sa oxidation reduction potential or, you know, the ability to reduce oxygen, to gamit oxygen. So, muna siya. cooked meat, medium, or broth. Okay, so those were the primary plating media. So again, some of them are specific ra gyud for anaerobes, no? But some of them kay medyo same ra sa ginagamit nato for routine culture media, naray gamay na modifications, okay? All right. Now, after primary plating media, we now go to inoculation procedures. Like, how do we inoculate the specimen? So for aspirates, of course, 1 drop of the purulent material or 2 to 3 drops if non-purulent. So when you say purulent, medyo baga, medyo thick, daghang plema or na mga mucus, no? Mucoid ba siya? So 1 drop ra kay medyo thick na siya. Pero if non-purulent, 2 to 3 drops lang. And MIS, 3 kraka to obtain well-isolated colonies. For tissue, di ba, homogenization or i-grind mo siya para madurog, mahimo siyang gagmay ng mga pieces para mas manageable pag-process, no? So after grinding or homogenization, like na yung grind, the suspension is used to inoculate the media. So as described earlier for aspirate. So kay liquid naman siya, medyo liquid na siya consistency, pwede na kag mo butang og 1 drop or 2 to 3 drops. Okay? Alright. Ayan. And for swabs, again, vortexing, removing the fluid from the swab. So, imo siyang ipatuyok gamay or imo siyang i-vortex para again makuha itong mga katong nitapot sa swab na mga kagaw or fluid. After that, remaining liquid, suspension, still the same. A drop, 1 drop or 2 to 3 drops. Pareha sa aspirates, ang imong ibutang sa culture media. Alright? Okay. Ayan. Now, next, we go now, of course, after mong inoculation, no, we now go to incubation. So, how do we incubate your anaerobic culture? So, generally good, ang general parang rule of thumb, yes, for anaerobic incubation is dapat wala dyan oxygen. Dapat wala dyan presence of any oxygen. Because again, your anaerobes do not want that. And ang anaerobes atong kailangan i-isolate, di ba? So, if na-ay oxygen, then malimit nilang growth, mamatay sila, so di ta maka-isolate o kagaw, okay? Alright. So first one is, again, you place the inoculated plates, rather, into the anaerobic environment at the temperature 35 to 37 degrees Celsius for at least 2 days or 48 hours. Again, cultures must not be exposed to oxygen until 48 hours of incubation. If a holding system like anaerobic jar ba is available, plates can be examined at 24 hours. So like na ay jar na pwede nimong niya anaerobic ang sulod ana niya na conditions, then pwede mo ibutang dito ang plates, okay? Alright. So, pwede na siya dito i-incubate. So, after 24 hours, pwede na siyang i-examine. Okay? And kung further incubation pag yun, or like wala pa na human imuhang testing, of course, ibalik mo siya sa imuhang anaerobic environment. Okay? And natay backup broth. Usually, example, nakatry ko sa amo sa Soto, CSF. When we receive CSF samples for culture, no, naadyo na backup broth na thio. Okay? Thio broth yun na, usually, ang CSF. So, or mga body fluids, mga sterile body fluids. Because we can expect na base na ay possible mga anaerobes from there. Okay? Of course, they are coming from your internal organs. They're coming from mga sterile sites, okay, that are deep, alright, far from the surface, na punog oxygen. So, munang mag-expect po na possible na anaerobic organism there. Okay? Alright, so again, backup broth natin is Thiogl Glycolic, alright, for 7 days. And if there is evidence of growth, subculture to BAP. So if walay, ang primary plates walay tubo, pero ang thio kay naay turbidity or naay tubo, so we can subculture that to BAP para ma-isolate na to, okay, or ma-identify. Alright. And again, Thiogl Glycolic broth is backup broth. It's also an enrichment broth para, again, if gamay pa ang pathogens na naasa mong specimen, iyasang padaghanon, alright, para mas easy, mas dali na lang siya ma-isolate. Okay. Alright. Ayan. And a final report of a negative culture is given after 7 days. So same siya sa imuhang blood culture after 7 days, no growth after 7 days of incubation. Pero for actinomyces, at least 10 days. So actinomyces is a branching filamentous, mura siyang nocardia, alright? Na mo branch, mura siyang fungus-like bacteria, actinomyces. Okay. Alright. Okay, now we now go to different incubation ng mga systems na ito pwede magamit. First is your anaerobic chamber. This is considered to be the ideal good na anaerobic incubation system because again, it provides oxygen-free environment for inoculation, incubation, up to work-up. So, tanan good from the start of inoculating to incubation to work-up, pag-identify nila as in oxygen-free good ang environment. Okay, so your requirement is, of course, dapat na i-catalyst. Ang catalyst ang mo remove sa residual O2 because the catalyst captures O2, okay, and combines it with, muna siya, wait, tama ba? Tama, ang catalyst ang mukuha sa O2 or oxygen from the atmosphere, mga residual, mga ginagmay from the chamber. And the desiccant is, siya mo absorb sa water. So what happens, the catalyst, no, it absorbs oxygen and combines it with hydrogen. Okay, and the formation of that is, of course, water. Now, kanang moisture na water is now created or is now captured by your desiccant, which is the silica gel. Okay, yes. A solid-state dehumidifier may be used as an alternative. So, ang point sa desiccant is, of course, yeah, it captures the water or the moisture na ma-create from the combination of the oxygen and hydrogen na gibuhat sa catalyst. Okay? Because again, we don't also want water. Alright? Kaya magbasa ang imong environment that could contaminate your plates. Okay. Alright. And oxidation reduction indicator, just an indicator kung taas pa ba ang oxygen level or naabay oxygen na naa dito. So once na ani na indicator na it indicates na naay oxygen, then of course, kailangan na matangtang to na oxygen or na mga appropriate measures to be done para mawala to na oxygen. Okay, so again, methylene blue or resazurin. Ayan. So na po yung mga digital oxygen indicators na pwedeng magamit. And of course, anaerobic gas, your gas can be hydrogen, carbon dioxide, or nitrogen. Basta walay oxygen. Okay? Okay, alright. Ayan. Okay, so here's an example of your anaerobic chamber using palladium-coated alumina pellets. Now, the palladium serves as your catalyst, di ba? So hydrogen, di ba? Nagpump makag hydrogen gas dito sa imuhang chamber. Alright, so if naay oxygen na available dito, na naay available na na-appeal sa imuhang anaerobic chamber, the catalyst, palladium catalyst, combines hydrogen na gipump from the external source, di ba? And oxygen to produce water. Okay, now this water is again captured now by your desiccant. Okay, all right, and can be inactivated by excess moisture and H2S kung ma-poison siya or if naay mga additional gases na na-appeal. Now, di ba, we don't want water because again, this excess moisture can inactivate your catalyst. So, dili na siya maka-convert nun sa oxygen if naapay na bilin na oxygen dito. Okay? So, muna siya yung purpose sa desiccant. That's why we need a desiccant to absorb the water that was created by this reaction. Okay? Alright, ayan. So, again, it can be reactivated after each use by heating in a dry heat oven for 1.5 to 2 hours. Okay. And we have also resazurin. If naay oxygen, it is color pink. But if wala, it's colorless. Okay. And so the same for methylene blue. If naay oxygen, blue siya. And of course, if wala, colorless. Or white, di ay white. Sorry. Okay. Ayan. So, nasa pangalan na. Kung resazurin, pink. Kung naay oxygen, if wala, kay colorless. Pero for methylene blue naay oxygen, color blue siya. And if wala, colorless. Okay. Alright. Ayan. Now, this is an example of an anaerobic chamber. So, glove box type. So, ang examiner or medtech, iyarang isulod yung kamot sa gloves. Okay? Tapos, kanidiri, mura siya dira na musulod ang specimen. Mura siya biosafety cabinet na level 3, na type 3. Katudjong glove chamber na naaray butanganan sa specimen. Okay? Ang difference lang ani is, ang environment is anaerobic. Walay oxygen as much as possible. So that's a glove box type of anaerobic chamber. And kani gloveless. Okay. So naa siya dissecting environment. Okay. So what happens is, still the same, isulod jud ang whole kamot sa patient. Pero, sa patient, sa medtech. Tapos, mag-wear siya own gloves niya. Alright? So, mura siya difference. Kani kay nanay gloves daan, kani kay murag naray opening na tabon na murag cover na tabon, okay? Then ang medtech gyud ang mag-provide siyang own gloves, okay? Maray difference. And naa siya dissecting microscope na attachment. So murag nanay microscope daan na included sa imuhang anaerobic chamber. Okay. All right. Ayan. Now, we also have what we call your anaerobic jar. So example, if wala mo'y katong anaerobic chamber kay mahal, so pwede po na ay jar. So one of that is the gas pack. So, mag-add po kag water to the gas generator to produce the gases aside from oxygen, like hydrogen, nitrogen ba, and ana. So, you add water to produce the gases, and the H2 reacts again with oxygen to, by the way of a palladium catalyst. Same sa ganina. Alright, so gas pack envelope plus water, it produces the necessary kanang gases na kailangan sa imuhang organisms, if capnophilic ba siya, and hydrogen also. And in the presence of residual O2, again, mo-combine siya with H2 by the presence of a palladium catalyst to produce water. And kanina water again, muna siya ang i-absorb sa desiccant. Okay. Alright, ayan. So this is an example of a jar. So jar gyud siya, alright? And kani siya ang murag ang muna siya ang dirigikan ang diri mo siya ang butangan og water, I think. Yeah. So muna siyang envelope, muna siyang i-open, I think. Tapos na siya yung water na i-add there. Not quite familiar with how it is operated, no? Pero yes, muna siya i-point na. That's gas pack according to Becton Dickinson. And this is the gas generating envelopes, di ba, nasa picture, and the methylene blue indicator. So katong ganina, muna siya ang gas envelope, okay? All right? And muna siya ang gas bar, ang jar mismo, ang rack, and ang kaning palladium catalyst na chamber. Okay, all right. That's the gas pack jar. Okay, all right. Next, we also have what we call the anoxomat, advanced instruments, no? Yeah, so mag-provide siya gases. So, it's in ani. So, as you can see, siya ang mag-provide sa gases sa imuhang jar. So, dili na ka mo gamit atong envelope, di na ka mo gamit atong palladium catalyst, kaya mo provide na siya og gas na aside from oxygen. So, yeah, within 3 cycles of evacuation and replacement gases. So, example, mag-supply siya og nitrogen. So, first cycle ko na pudto, replace na puglain na gas, parang inana. So, na evacuation replacement cycles, 3 cycles of that. So, that's anoxomat. Okay, all right. Ayan. And next, we also have anaerobic bags. Okay, so example for transport, or if gagmay ra imong specimens or plates, all right? So, 1 or 2 ra ang plates isulod sa bag na oxygen-free or gi-remove ang oxygen, and the bag is sealed and i-incubate siya at normal incubators na to. Okay, example like this. Ayan. So, isulod rin nimo ang plates nimo, usually gagmay ra if duha ka buok, all right? And then incubate at your normal incubator. Okay? As long as ang iyahang environment is anaerobic, walay oxygen. All right. Okay. All right. Now for the next part of the video, we will now go to the indications as to or mga clues na makaingon na ang imuhang gi-isolate or ang imuhang tubo sa culture kay could be an anaerobe. Okay? So that's what we're going to discuss until the end na. Hopefully last video na to ang next sa atong anaerobic culture. Okay? All right.